Good afternoon, everyone. Welcome to Sensory Spring 2021 conference series. This is our last seminar presentation this semester. And I'm Aminur, moderator for Sensory conference series. The Sensory is a multidisciplinary group of researchers with interests in sensor materials, method, devices, and sensor applications for healthcare, environment, civil, petroleum, chemical industries, and military applications. This group provides an opportunity to exchange ideas, present early findings, and explore collaboration and interdisciplinary funding opportunities. Sensoric's aims are to connect sensor technologies, middleware, and applications, and to provide an interface between academic and industrial research. So, before giving the floor to our guest Yaoran Liu, I would like to express my invitation to all interested personnel to join Sensoric's committee as an executive members. We would be very happy to have you in our team, and Sensory gives you an opportunity to utilize your skills and make a network with other interested personnel. So here I have, you can see our YouTube channel and Sensory Research Interest Group. Today our guest is Yaoran Liu. He is a PhD student in Electrical and Computer Engineering Department. He is working with Dr. Yu Benzi. He did his bachelor from University of Electronics, Science and Technology of China. And his research interest includes cytopectral measurement for print of care testing of chiral biomarkers of diseases and complications. So let us give the floor to Yaoran. And if you have any questions regarding the presentations or anything, please feel free to ask after the presentation. Thank you.
So I think everyone can see my screen. So first of all, thanks for the brief introduction. I'm Yaoran Liu. I'm from Dr. Yue Binzhen's group. Today, I'll present our recent research related to the plasmonic chiral sensing of biomarker in human bodies for chronic disease monitoring. I'm from Department of Electrical and Computer Engineering. I'm a 6 years graduate student. So let me get out the pointer. Okay. So first of all, what the chirality? So chirality refers to objects that cannot superimpose on its mirror image. Take your hands, for example. Your right hand cannot superimpose with your left hand. Chirality can happen in a lot of subjects, such as chemistry, biology, and mathematics. So for chiral molecules, its center is typically carbon atoms with 4 different groups. Chiral molecules is extremely important for the pharmaceutical industry. Take this thalidomide drug, for example. For its right-hand structures, it typically used for nausea relief drug. However, for its opposite-handed structures, referring to left-hand thalidomide molecules, it will cause a severe teratogenic disease for the children. So it's very important to distinguish the hardness of these chiral molecules for the pharmaceutical industry. Not only is it vital in the pharmaceutical industry, but also chirality happens in human bodies. For normal humans, we typically only have one type of enantiomers in our body, referring to biological homochirality. For example, we only have L amino acid in our bodies. We will not have D amino acid in our bodies. However, there's an increasing research revealing that humans with chronic disease will have 2 types of amino acid in their bodies. So why chronic disease is so important in the recent years? It is because the chronic disease is rapidly increased in the past years. For example, around 50% of the total population in the United States have a chronic disease. And also, chronic disease are related to other kind of disease, such as COVID-19. Study found out that patients with chronic disease will have a higher possibility to be hospitalized than those who don't have chronic disease. Therefore, it's extremely important to get diagnosis or even prognostic if you have chronic disease. So what's the relationship between the Chiral biomarker and the chronic disease? Here I list some of the selected biomarkers which are related to the chronic disease. As you can see, if you have a chronic kidney disease, simply I put like a KD here, diabetes or some like Alzheimer's disease, you will have some sort of like elevated D-enantiomers in your bodies. This can be some of the D-amino acid or like a D-lactate. There's several conventional methods to detect the Chiral biomarkers in biofluidics. This includes circular dichroism spectroscopy, polarimeter, chiral liquid gas chromatography, chiral nuclear magnetic resonance, and fluorometric assay kit. However, they all have their own limitations, such as high sample volume and high analyzed concentrations. Some of them even require a tedious preparation steps, such as chiral derivatization agent or chiral stationary phase. In order to overcome the limitations of the traditional method, in our group, we develop a new type of metamaterials called more chiral metal surface, simply say MCMs, to detect the handiness of the chiral molecules in low quantity and low volume. So as shown in these schematics, our MCMs consist of two layers of gold nanohole arrays stacked with each other. By properly tuning the rotation angles between these two layers, we can either form a left-handed MCMs or right-handed MCMs. Depends on the relative rotation angles between these two gold nanohole structures. And as you can see, we can fabricate this metal surface in a large areas manner. We further simulate and measure the circular dichroism spectra of these types of MCMs. As you can see, they show the opposite circular dichroism spectra. For the left-hand MCMs, it shows like a more positive values in the circular dichroism spectra, while for the right-hand MCMs, it shows the opposite spectra. By properly tuning the rotation angle between these two layers in like 10 degrees, 15 degrees, and 20 degrees, we can tune the its circular dichroism spectra. Using these two pairs of MCMs, left-hand MCMs and right-hand MCMs, we can use them to detect the handiness of the chiral molecules. As the first demonstrations, we try to detect the handiness of chiral proteins, such as CON-A proteins here. We calculate the symmetry factors, delta, delta, lambda. As you can see, we get the positive values here, indicating a more right-handedness properties of these proteins. Then we try to use these metamaterials to detect the chirality of small molecules, such as chiral drugs. For its right-handed cyanidamide, we get a more positive values in delta, delta, lambda, indicating a more right-handedness properties for this kind of molecules. While for its opposite enantiomers, we get the negative values, indicating its left-handed structures. Currently, our concentrations using these techniques is around 10 millimolars. However, this cannot meet the requirement to detect the chiral molecules in a psychological environment because, for example, the serums or glucose in a human body is typically below 10 millimolars. Therefore, how can we improve the detection limit and the sensitivities of our current platform? It's worth to note that our current platform is based on a diffusion techniques. So to overcome these diffusion limited biosensors, in our group, we developed a kind of a concentration technique. Briefly, we try to bring the molecules in the liquid into a localized areas so that we can increase the local concentrations of molecules to improve the sensitivity. The basic principles of this technique is using a bubble. So basically, you focus a laser beam on a plasmonic substrate and generate macro bubbles on the substrate. The macro bubble will generate further generate a Marangoni flow. This Marangoni convection flow will efficiently bring and drag the molecules in the solutions closer to the laser point here. So we successfully achieve printing and concentrations of polystyrene beads with different sizes and even fluorescence quantum dot. Then I'm thinking about whether we can apply this technique to print and concentrate biomolecules for the biosensing with improved sensitivities. So here, as the first demonstrations, I try to detect the hardness of the glucose. Here, the concentrations of the metabolites in the solutions is around 100 micromolars. Then I apply 3 consecutive microbubble concentrations. The duration of each bubble is around 5 seconds. So we measure the circular dichroism spectra before and after the microbubble concentrations. For this black line, this indicates the circular dichroism spectra before the bubble concentrations. The red, blue, and pink curve represent bubble 1, bubble 2, and bubble 3. Each represent the bubble concentrations. So if we compare the circular dichroism spectral change before and after concentrations, we found that for L-glucose, the left-hand MCMs will show a continuously red shift, while for the right-hand MCMs, it show a continuously blue shift. For the D-glucose, for both left-hand MCMs and right-hand MCMs, they show opposite spectral shift. We further calculate the dissymmetry factor, as I mentioned before. It's referring to the spectral shift of the right-hand MCMs minus the spectral shift of the left-hand MCMs. We get the negative values, while for the D-glucose, we get the positive values, indicating we successfully differentiate the hardness of the glucose. Then we try to find out the detection limit using the bubble concentration technique. We found out that our detection limit can be as low as 100 picomolars, compared with which is 7th orders enhancement than the case without bubble. Then we try to apply this technique directly to detect the biomarkers in a human urines. So the protocol is that we collect the fresh human urines in a human body or a mice, followed by the centrifuge. Then we take the upper layers of the urine and redisperse it into our microfluidic devices. Then we apply microbubble concentrations. So these molecules or metabolites in the urine will efficiently get printed or concentrated near our plasmonic substrate. After the bubble collapse, the molecules stay there with high concentrations. Then we measure the circular dichroism spectra before and after the concentrations. As you can see, for the dissymmetry factor, its values show is approximately zeros for normal humans. However, for the diabetic patients, we get more positive values in the dissymmetry factors, indicating there would be more right-handed molecules in the diabetic patients. And we further calculate the conduct the ROC analysis and calculate the area under the curve, which give you the diagnostic accuracy. So we found out that using our technique, we get the diagnostic accuracy around 84%. Compared with the conventional technique using the glucose meter, we show a higher values here. So as a conclusion, we develop a more chiral metal surface for detecting chiral molecules. And we observe diabetes-induced abnormal chiralities of metabolites in human urines with detection accuracy around of 84%. We believe our platform will benefit the development of palliative care devices for first-line non-invasive screening of chronic disease. So finally, I'll thank my advisor, Dr. Yuebing Zheng, and also our collaborator from UT Health Center in San Antonio, who provide the clinical samples to us. And also thanks for the organizer and the participant today. And thank you for your questions.
Thank you, Yaoran, for your amazing presentation.
Yeah.
And this is like really, really interesting. And I think we should give floor to the audiences to ask any questions.
Yeah. So first, I think of from Dr. Hunter, right? Yeah. So yeah, I make most. So actually, we make these images by ourselves. But for some of the cover images, we make it in a group. So for this kind of image, I think 2-person draw it. Yeah, yeah. But for this data, we plotted by ourselves. Yes.
Hi, Yaoran.
Yeah.
Thanks for the great talk. May I ask, when you do the measurements, because we know in the biofluids there are many complex, very complex molecules, right? You said there are many chiral molecules inside. So
yeah,
will the other chiral molecules affect your measurements when you're targeting at specific diseases?
Yeah, it's a really good question, sir. So basically, currently we haven't do any separations. So we just measure sort of like an overall chirality in the urines. So as I show here, so we get the more positive values of I mean in the DCM trafficker. As I said before, it's indicating a more right-handedness of the molecules in the solutions. This can be a glucose. It can also be the amino acid, as I mentioned before. So currently we are doing like a separation technique. So currently we are doing like a LC separation, which is liquid chromatography. And also we also conduct a capillary electrophoresis. So basically we try to separate the molecules with different retention time so that we can specifically target a specific biomarkers in the fluidics to detect their chiralities. And eventually we are thinking to combine our MCMs with the chromatography to integrate the whole components like LCMS for the chirality profiling. So that's it. Thanks.
Yeah, thank you.
Is there any more questions? Yeah, I have one question. What instrument do you use to measure the concentration of the molecules after the collapse of the bubble?
So it's kind of like maybe I missed some details here. So as you can see, I mentioned many times of the circular dichroism, which simply see as a CD. So the CD can be calculated by the absorption difference between the the left-hand circular parameter and right-hand circular parameter. To get this curve, we have our microscope equipped with a spectroscopy. So basically, it can measure the absorption and the transmissions of the localized areas. So something like a detector, but it's measured the intensities there. I don't know whether you guys are familiar with SPP or, detection or plasmonic sensing detections using like absorption or transmission. Or
yeah,
something like that, yeah.
So from our audiences, are they interested in asking any questions? Yeah, I think we're okay now. And thank you, Yaoran, and our audiences for their valuable time.
Yeah,
and this was like really a nice presentation. And with these presentations, we are closing our seminar talk for this semester. And everyone, please feel free to send any comments or suggestions regarding sensor rigs in sensors_rigs_students at utilis.utexas.edu. And also follow us on our Twitter. Our Twitter handle is also SensorRig, and we are the only SensorRig in Twitter. And thank you, everyone. Hopefully, see you in the next semester.
Thank you all, guys, and take care.
Thank you.
Thank you. Yeah.